So today we'll be talking about mapping the cancer genome, its history, its background, what we've done so far, and where we're going to go from here. So our discussion about the history of genes in cancer and how they're related begins a little over a century ago in 1890. Uh, in that year, David von Hansmann, a German pathologist, detailed the mitotic structures of 13 different carcinoma samples. And he noted that every one of them contained aberrant characteristics, like unequal distribution of chromosomes during the anaphase of mitosis. At the time, this discovery was largely ignored and wouldn't be picked up for another decade. Then, in 1902, Theodor Boveri, a German biologist, decided to investigate this link between abnormal chromosomes and malignant tumors. He discovered that the abnormal distribution of chromosomes that von Hansmann had found during mitosis meant that there was an unequal distribution of inherited factors, which usually hurt the individual. In some cases, this led to uncontrolled growth of the malignant cells, which would pass its abnormalities onto its daughter cells. Now, his studies set up the foundations of genetic research on cancer and predicted the existence of tumor suppressor genes, oncogenes, and even predicted how effective treatments like radiation therapy or chemotherapy might be at killing these cells. The next major milestone in genetics research occurred in 1960 when researchers discovered the Philadelphia chromosome, a product of translocation between chromosome 9 and 22. When this happens, parts of the two different chromosomes switch, as you can see in the figure, and it leads to abnormal chromosomes. Now, this was important because this was the first genetic defect associated with a specific cancer, and this finding eventually led to the identification of other chromosomal translocations and the discovery of other oncogenes. The next two major milestones were discovery of tumor suppressor genes. So in 1979, P53, which was at the time thought to be an oncogene, was discovered. It is a tumor suppressor gene and the most frequently mutated gene in human cancer. Now, this, along with RB1, a retinoblastoma gene, were two tumor suppressor genes that were found to be very important in cancer. And understanding how malignant tumors shut down their tumor suppressing abilities was very critical to research that developed in the future. Now, one of the biggest steps in cancer research was the Human Genome Project, which began in 1990. Now, because of all these discoveries being made about cancer at a genetic level, there was an increasingly larger push to have a larger scale project investigating this disease at a genetic and genomic level. And so in response to that push, the Human Genome Project, which began in 1990 and completed in 2003, was established to lay the foundation for this research. Now, in addition to giving us sequences of 3 billion base pairs of the entire human genome, it allowed researchers over the 13 years that it operated to optimize technology, make it more efficient. Now, this optimized technology led to cheaper technology. For instance, at the beginning of this project, it cost around $10 to sequence a single nucleotide base. Now, it costs less than a penny. Now, the Cancer Genome Atlas started in 2005 under the supervision of the NCI and the National Human Genome Research Institute. And its goal was to compare DNA sequences and other characteristics of normal cells to cancer cells to figure out what the differences were between the two and which differences caused what are known as the hallmarks of cancer. Now, these hallmarks, these 6 abnormal abilities that cancer cells have, allow them to override the controls that are built into the body and spread through the body in a lethal manner. One of the most important characteristics of cancer cells is known as sustained proliferative signaling. What this means is that they can grow using outside signals that normal cells may not have. So normal tissues are able to control the production and release of these growth factors. Cancer cells lack this regulation, and so what they can do is produce their own growth factors or signal nearby cells to supply them with growth factors, which causes the overproduction of cancerous cells that we see. Now, in addition to sustaining this growth, cancer cells must also be able to bypass the programs that are built into our body that prevent cell proliferation when it's not needed. And many of these programs depend on what we call tumor suppressor genes. Two tumor suppressor genes mentioned earlier are RB1 and P53, both of which play an important role in shutting down unnecessary cell growth. RB1 takes in signals from both inside and outside the cell to decide whether the cell should proceed to its growth and division phase. If a cell has a defective RB1 gene, then that cell is missing an important regulator that, when absent, will allow for uncontrolled cell growth and division. Similarly, a P53 gene monitors the cell's internal environment for any abnormalities. If it detects something unusual, then P53 can pause the cell's growth until this problem has been resolved. In the event of irreparable damage that can't be resolved, then it can also trigger apoptosis, also known as programmed cell death. A lack of these two regulators is very characteristic of many in the cancers we see. Another hallmark ability is cancer's metastasis, which is its uncontrolled spread through the body. Now, this is normally prevented, but cancer cells are able to avoid and ignore the body's built-in mechanisms which prevent cancer from traveling to other tissues. This facet of cancer is complicated and can lead to the worsening of a patient's condition. Cancer cells also bypass the inherent limitations of cell growth. They have nearly unlimited potential to replicate and grow, whereas normal cells can only go through a certain number of growth and division cycles. The fifth hallmark ability is known as inducing angiogenesis, which refers to the development of new blood cells that cancer can promote, which provides them with the oxygen and nutrients they need to continue growing and replicating and spreading through the body. As mentioned earlier, another characteristic sign of a cancer cell is the ability to resist apoptosis, programmed cell death. This is the idea that there are mechanisms that should trigger or carry out a self-destructive program in damaged cells, but in cancer cells, these programs are disabled or overridden. One example mentioned earlier was the P53 gene, which is inactivated and defective in cancer cells. Now, the question you must always ask yourself is the research you're doing beneficial? Is it going to help someone? Having enormous information and data is necessary, but simply having it doesn't tell you whether you can use it to help cancer patients. Now, genomics research looks at the differences between normal cells and malignant tumors and looks to target the hallmarks of cancer we discussed on the last slide. Now, thankfully, previous studies have looked at cancer at a genomic level prior to the implementation of the Genome Atlas, and these studies have resulted in important discoveries that led to better treatments. First is the study from the Sanger Institute that began in 2001. The Sanger study sequenced about 20 genes in 378 cancer samples, and they found that the BRAF gene was mutated in about 70% of the malignant melanoma cases they looked at. Research since then has tried to block and reduce its gene activity, inhibit MEK, which is a protein that becomes overproduced from the BRAF mutation. Eventually, research led to the development of treatments that went to clinical trials. These clinical trials produced two drugs, dabrafenib and vemurafenib. Recently, the FDA has approved these drugs as treatment for late-stage melanoma. Both drugs inhibit an enzyme associated with BRAF, allowing for treatment of its cancerous activity. Another study, which took place on an even larger scale, was done at Johns Hopkins, which sequenced 13,000 genes from colorectal and breast cancer patients. And they discovered almost 200 different genes with significant mutations. They discovered mutations in genes that were involved in cell motility, signal transduction, transcriptional regulation, transport, intracellular trafficking, and many other mechanisms. These genomic studies have produced other treatments as well. Gleevec is the first therapy to target a known gene-based cause of cancer and was approved by the FDA in 2001. This inhibits an enzyme produced by a mutant fused version of two genes, BCR and ABL, that causes chronic myelogenous leukemia, as you can see in the figure, while a substrate would normally bind to the mutated domain. Gleevec competitively inhibits this binding and, by doing so, prevents this proliferation of the tumor cell. Gleevec is also effective against other diseases like gastrointestinal stromal tumors and other relatively rare cancers that involve similar enzymes. In addition to this, Herceptin has been developed, an antibody that interferes with HER2 receptors and is targeted for therapy in around 30% of breast cancer cases. Iressa and Tarceva are drugs that have also been developed, and they inhibit epidermal growth factor receptors, EGRFs. Normally, the stimulation of these receptors by growth factors outside the cell transmits signals to additional proteins and genes, which ultimately prompts the cell to grow by dividing. By inhibiting these receptors, you are preventing proliferation and growth of these tumor cells. These two drugs are especially effective in lung cancers. So what has the TCGA been doing? Well, under the supervision of the NCI and the National Human Genome Research Institute, multiple teams were picked in 2006, given a $100 million budget, and were told to investigate the genomic changes in glioblastomas in the brain, lung, and ovarian cancer. Among the reasons that these cancers were picked is that they account for around 200,000 cancer cases in the US and around 190,000 deaths in just 2006 alone. Now, there are four major elements to the Cancer Genome Atlas project: the Biospecimen Core Resources, Cancer Genome Characterization Centers, Genome Sequencing Centers, and the Data Coordination Centers. Specimens go to the Biospecimen Core Research, which then coordinates with the other four major parts of the project to uncover the large-scale genomic changes that contribute to the development and the progression of cancer. These centers will look for things like chromosome rearrangements, changes in gene copies, and epigenetic changes. Once these areas of interest are located, the genome sequencing centers will target them. It is estimated that 2,000 genes will be sequenced by 2014. It should be noted that even this large-scale research has its challenges. Because of the volume of information that the atlas will be processing, it will be difficult to separate irrelevant mutations from the malignant ones. Additionally, there may not even be significant overlap of mutations between cancers, and there might even be variety in mutations between patients for the same cancer. Once cells become cancerous, they can mutate at an even greater rate as their self-control and repair mechanisms fail, which will make it difficult to pin down which mutations are important. Since it began, the Genome Atlas has achieved a lot. In 2009, Phase 2 began, which intended to complete the characterization sequence analysis of 20 to 25 tumors by 2014. It has since then surpassed this goal, characterizing 33 cancer types and 10 rare cancer types. All of the published literature is publicly available and has aided the efforts of researchers in independent teams to increase the general knowledge about cancer and its genome. It has allowed us to develop new ways to classify tumors and developed new therapeutic targets so that we can treat patients better. The Cancer Genome Atlas has resulted in enormous benefit to the scientific community as it set the standards for investigating cancer and its genomic origins. Going forward, it will continue to develop its database and collaborate with other organizations to fulfill the original goal: to improve our ability to diagnose, treat, and prevent cancer.